organic. It's introducing the things that we're going to talk about. We have a lot of pieces to build here, and you're not going to put the whole together till we get through that first 4, 5, 6 chapters, and then we start getting into, you know, the methods of testing. But the immunology basics and the function of the immune system is better given in pieces. So this kind of goes and gives like a real overview of everything that we are gonna hit. So when you start looking at immunology and the history, you know, I'm always incredibly fascinated with how we discover things and how you know, history writes itself when it comes to medicine. So, you know, oops, getting really loud with a big truck. I will wait to talk because that's probably really loud for you. I'm lucky enough that my entire neighborhood, there are 1, 2, 3 houses that surround me on 3 sides, and they're all under construction. I'm super happy about that. Not really. So when we look at the history of medicine, the history of immunology, you know, it was trial and error. They randomly got exposed to things and figured out things from it. So for example, when you go back to Edward Jenner, okay, does anybody know who Edward Jenner is? You might not. No? So he did the smallpox vaccination. So he's the one that invented this. And how did he invent it? Well, number one, he tested it on himself. Talk about dedication. But he noticed that when milkmaids were milking cows, they would get like gross pustules all over their hands, but none of them ever got sick with smallpox. They got cowpox, but the cowpox was so similar to smallpox that their immune system developed antibodies against it and they fought off smallpox. So they never actually got smallpox. So it was really cool to kind of see this in action where he has, like, he was not an immunologist. He was like a zoologist or something. He drew birds and animals and things like that. But in his observations, he noticed something and researched it. Now, obviously, we can't just do things like this, you know, and sign up and say, hey, guys, you know, you don't know me for a hole in the wall. I have no qualifications. But yeah, come here. Let me test a vaccine on you. We can't do that now. But, you know, this is how we got to where we are today. You know, and I mean, I know I've told you guys stories about infection control starting in ancient Egypt where they utilized minerals and things to protect their eyes. It wasn't eye makeup, it wasn't vanity, it was protective so they wouldn't get sick, you know, from sandstorms, you know, tearing up their eyes or the side of their eyes and getting bacterial infections that way. So we really started with all of this type of research. And as you know, everything has a Latin base. So Latin itself, immunis, means basically protected from. So that's where the English word comes, immunity. It means we are protected from something, multiple things. You who knows? All right. Whoops. So this, I'm obviously not going to read through this. You can read it, but you're looking at down here, you know, you Chinese, again, playing around with preventing smallpox. They had no idea what they were doing at the time. Jenner, your cowpox vaccination. Edward Koch looked at delayed hypersensitivity with TB. Louis Pasteur, you guys all know Louis Pasteur. What did he do? Besides
aseptic technique.
Yes. Oh, absolutely. You know, down to Bucknam, Woodall, which is looking at that first test for typhoid fever. So all of these guys had a huge influence in history itself. So now you obviously, I'm not going to get up on my vaccine soapbox. You know, I have thoughts, but the ethical educator lets you get there by yourself. But this is going back to 2004, okay? We'll look at pre-vaccine cases and post-vaccine cases and the amount that were here. And sadly enough, these post-vaccine numbers are not changing for the better because of anti-vaccination has taken it to the nth degree. Because think back in 2004. What were you doing in 2004? Was there Facebook? Was there Instagram? I don't know if there was Twitter. I know there was MySpace, but you know, I had a flip phone back in 2004. Social media didn't exist. So now we're at this point where, oh, I am lecturing, beat it. Yes, this is how my life is going to go. And I am sorry that my son is out here interrupting me, showing me a picture of his alphabet soup that he spelled his name in. Can you please go back inside now? I said now. I apologize. I can't win. I'm going to actually start punishing if we keep doing this. There you go. But we're at this point where, you know, we're getting better and we're moving forward, but yet we're taking 10 steps back in history. So this is just a list of Nobel Prizes and what they've done, anywhere from serum antitoxins all the way down to monoclonal antibodies. And we've gotten to the point where we can use these monoclonal antibodies and then some. You know, we can use interferons, we can use these cytokines for treatment of cancers, treatment of autoimmune disorders. So we have lots of treatments that come from, you know, the early 1900s and back further, all this research that these individuals did. So this is an interesting theory, okay? So this theory, by the way, is not correct. It's the basis of what we have today, but it started this initial exploration in how we have this protection. So this guy, Paul Ehrlich, proposed this side chain theory. So basically, the idea that we had cells somewhere in our body that made these side chains on the surface of the cell, okay? And these basically were our antibodies, okay? And we had receptors for antigens that were on the surface of bacteria, and we would see binding of that side chain to that bacteria and caused a synthesis of new side chains that were finally liberated into the body as antibodies, as lock and key. So it's not 100%, but that's where we got our basics from. We got our basic antibody antigen reaction from this guy's research, or this guy's theory. And then we moved into von Behring and Catatso. They looked at serum. That's again, the liquid portion of your blood. They took that from animals that were immunized to diphtheria, and they could actually transfer that immune state to the unimmunized animals. So they basically took serum from an animal that had protectivity or protective antibodies to diphtheria, not that they knew what that was at that point, and then they put it into an animal that didn't, and then the animal got it. So basically, they came up with the idea of humoral antibodies from this theory as like the humors, okay? So remember, I think I talked to you about this either in phlebotomy or in urinalysis, where we had these good humors versus bad humors. Everybody based their theories on humors, and the health of the body was based on, you know, how many good humors you had. And if you had bad humors, you were probably ill and you needed to be bloodlet and to get out the bad humors. So all of this was based on this type of idea, where now we're seeing immunity coming with the humor or the liquid. So now we talk about it as the humoral immunity. Another thing they looked at was serum antitoxin. So basically, this theory where apparently this antitoxin, now again, I'll get to it, but this serum antitoxin, they thought neutralized different toxins, you know, killed off bacteria, caused agglutination, and it had all these parts in it. Turns out that it was really the antibody that did all of this. So this serum antitoxin was their first theory of an antibody. They just didn't call it that. And this is, you know, looking at this and going back and seeing that and seeing where we are today, you know, and what we know about the immune system, and even what we don't know. We still don't know a lot. We still discover new pieces and new things that the body is doing in the immune system every day. So then another guy came around, Mechnikov, I can always say that wrong. He had this idea that we had phagocytes, and these were cells that could engulf microorganisms. So basically, these were more active in immunized animals And so they thought that this was a major effector cell of immunity. And so again, your PMNs, your polymorphonuclear leukocytes, or your neutrophils, that's what they called the circulating phagocytes at the time. And then the fixed ones were called the macrophages. And we still use that terminology today, because you'll notice, I don't know if you checked out your lab stations at all, but the lab stations have the diff counters. You might not have known what it was, or if you've been in a lab before, you know what it is. But basically these diff counters, some of them still have PMNs on the counter for the neutrophils. That's how old they are. So this is just an example of how our immune system will respond to an invading bacteria. And we'll talk a lot about this when we talk about the function of white blood cells, where we're having this entrance of the foreign entity. Quick question, can you see my pointer when I circle it?
Yes.
Like,
Yes.
do you see my pointer?
Yes.
Okay, I'm not going to keep circling things. That's why. Okay, so you see this foreign invader, and what's going to happen is our body detects this foreign invader. And so what will happen is you'll see our white cells marginate, and we'll talk about this as we talk about white cell development, but most of our white blood cells, or our neutrophils, will marginate along the edges of the capillary because they're ready to respond. They're ready to do their job. So you'll have some free floating, and then we'll have some of the portion marginating. That's why you're not supposed to do this when you draw blood, because you can falsely elevate the white blood cell count because you're releasing those marginated white blood cells off the edges of the capillaries and the veins. So as a process of extravasation occurs, we're going to have all kinds of chemical responses and things like that, where your complement, your antibody production, C-reactive proteins. We're going to see all of this. We'll see cytokines. We'll see all sorts of your cytokines or crosstalk chemicals between your cells to create this response, which we will talk about when we talk about the cellular function within the immune system. So in this whole idea, we called this diapedesis, or that crossing, that extravasation of the white cells through the capillary walls, or diapedesis, allowed these cells to go out into the tissues and to be responding. So then we started seeing a fight between the concept of humoral immunity and cell-mediated immunity. Which one is it? So you started seeing this mix-up. Along comes research in the 50s. So remember, we did a lot in the 50s where we started to, having the technology to be able to identify things a little better. So we discovered this thing called the lymphocyte, okay? And this was the cell that they discovered was responsible for cellular and humoral immunity. So basically the main functions of that immune system were defense, homeostasis, and surveillance. So I always picture the lymphocytes as our police officers of our body, okay? They are surveilling the area. They're looking for parking tickets. They're looking for speeding tickets. They're always there. And our lymphatic system, I always view as basically like a separate highway that goes along our regular average Joe highways. And the separate highway allows for the passing of our police officers faster. So if you've ever taken the Hove lane in Boston, I think about it as like the lymphatic system is the bypass, so we can go wherever we want. So when we look at, defense, obviously our main function, we want to defend against any type of invading substances. If it's foreign, we want to fight it off. The homeostasis, part of our function as well is to keep us balanced. We can't just be full of dead cells that don't work anymore. So we have programmed cell death, or apoptosis, as once a cell has reached its maturity level or has become problematic, programmed cell death will occur, and our macrophages will come behind and they will recycle any of the leftover cellular material. So iron will go, to either storage or usage of blood, red cell production. Oh, and of course now someone's vacuuming. And then the proteins will get broken down to the amino acids and brought back to the liver to make more proteins. So things like that. And then surveillance, obviously we want to precipitate and destroy any mutant cells. So our immune system is really good at, detecting foreign entities as they survey. And it's basically like a little hey, this cell bounces off. Okay, check, you're okay. Oh, you're okay. Ooh, wait a minute, you're not okay. And they're going to trigger that cell is a dying cell and we need to get rid of it. And we'll talk more about this process as well, so don't panic. I love immunology. As you can tell, I get really excited about it, but I know this is a lot in one day. So as we're going over this overview, we'll break out all of these pieces as we get further into the immune chapters. So we have different types of immunity, okay? So when we look at our natural or innate immunity, so this is basically we're able to resist infection by stuff that's normally present, okay? So for example, we don't need to be prior exposed to anything, but our skin is always going to be a barrier. It's always going to have natural flora, although I don't know. I'm worried about our overuse of hand sanitizer. We're like wiping out our normal flora. But the idea is the normal flora on your skin is actually protective because your whole body is covered with that bacteria. If there is a foreign bacteria that's trying to gain entry into the body, number one, our bacteria is going to block up those receptor sites and it's going to utilize the food sources. So the idea is there'll be no place for the foreign bacteria to bind and there'll be nothing for it to consume. So that's our first initial protectivity. You can look at all different things where no matter how you are exposed to it, okay, you could be exposed to different things over and over and over again. The response is always going to be the same. It is never going to be different. So we can see our immunity either being innate or adapted. So our innate is basically we're born and it's independent again to that exposure. So we will see things like our activities of our phagocytic cells. It's always going to respond to a foreign invader. That's its job. And then we can see, antimicrobial substances, metabolic changes in the body. So the pH of the vaginal tract, the pH of the stomach, things like that. All of that is there for us and it's not going to change. And basically, this immunity itself is going to include immunity that causes, one animal species to be resistant to microorganisms affecting other species. So, for example, we don't get distemper. Dogs do. We don't. Where things get scary, as you can imagine, is when we start looking at mutant viruses like COVID. Where it didn't originate as a human virus. So things like this, can be scary. Sorry, my throat's getting dry. Of course, Diet Coke's probably not going to help that. So here's an example. Can you see this full screen or is it just mine that's got something in between? Can you see all of this?
Yeah.
Okay, so here your innate immunity. So this is a rapid response, okay? Because remember, this is our battalion that we have. It's ready to go at all times and we'll treat everything exactly the same. So your mast cells, your dendritic cells, macrophages, natural killers, complement, basals, eos, newts, granulocytes, okay? All of those are going to respond as normal. So our adaptive immunity is a little different. So this adapted is going to be slower because our body has to respond to it. Our body is going to respond to a different entity differently than another one. And then middle a little bit. So, whoops, I'm frozen. All right, can you hear me?
Yeah.
Okay, so the types of immunity that we see, okay, so you're adaptive or you're acquired. This is where we basically have a specificity to individual foreign entities, whether it's, a different type of pathogen, microbial agent, anything. So basically, we will remember if we've been exposed before. So for example, every one of you has a hepatitis B vaccination. You all have antibodies to hepatitis B. So you are going to remember that you got that vaccine. So you are going to produce more antibodies if you get exposed to hepatitis B again. So basically this adaptive immunity will result after you've been exposed to microorganisms and other foreign substances. We even get funky ones where our naturally occurring blood antibodies to our blood types. Okay, so I'm type A. I have naturally occurring antibody against B antigen cells. I know B cells are foreign. How did my body learn that? Well, the theory is that we were exposed to something that looks like the B antigen on the red cell, most likely a pathogen, and we've mounted an antibody response to it. So unfortunately, if I were ever to be transfused with B blood, I am going to attack that antigen because of that essentially naturally occurring antibody. It's really fascinating, and we'll talk more about that as we get into, the ABO RH systems. So our immune system will actually respond to these microorganisms by making our antibodies, and our lymphocytes will actually act against these foreign invaders. So that's our naturally active immunity. So our passive immunity, this is basically a transfer of antibodies from one person to another, and typically, again, from no intervention, it's natural. So most of these are our antibodies that come from our mom. Not all antibodies cross the placenta, and when we talk about antibodies, we'll discuss which ones do and which one don't. But a lot of protective immunity for that newborn comes from the mom, and it crosses the placenta. They will, because the baby is not making its own, because it hasn't been exposed to any of that, those will disappear after the first couple months. And you'll notice that that's where we start staggering all of the vaccinations so that baby develops their own antibodies. That's why when your kids first go to daycare, they get sick and you get sick all the time, because they're being exposed to all these new things and they're bringing it home to you. Hold on. So our artificially acquired active immunity is basically we deliberately do it. So vaccination is the most common way that we do this. And so most of these are preps of our foreign substances, so killed microorganisms, virus, live attenuated organisms, so they're still alive, but their infective capabilities have been removed, or using inactivated toxins. So for example, when we initially had that polio vaccination, we had a killed and we had a live attenuated version. And we'll see different versions of these vaccinations throughout our lifetimes. So I like, me, I like my charts. So we have a comparison chart of innate versus adaptive immunity. So innate can take just a few hours. It's limited, it's fixed, it's always going to be the same, and it's identical to the first time you respond to it. Our adaptive takes a little longer, it's very diverse, and basically we are going to see a really more rapid response as we see subsequent exposures, because the more we're exposed to it, the more antibody we're going to develop, and then we'll act faster and be able to defend that. So here's an example of what is specifically characteristic of innate versus adaptive. So you'll notice here, adaptive immunity is not inherited in a genome. It's not expressed by all cells. It doesn't trigger an immediate response, and it doesn't recognize broad classes of pathogen. It recognizes that one specific pathogen. And then you'll look over here for your innate, and basically it does not have multiple gene segment, it doesn't require, and it doesn't recognize a wide variety of molecular structures. So these are the major differences between innate and adaptive immunity. So when we talk about like our natural defense mechanisms, okay, so we have external systems, so attempts that, you know, basically we can prevent entry of pathogens. So we already talked about one of those with the skin. And then we have the internal system. We have a response that's generic if some foreign entity enters our body. And both of these external and internal systems will promote that phagocytic activity. So again, we talked about anatomic barriers, so we talked about the skin. Mucous membranes also do that. Physiological barriers, we already talked about pH, but we didn't talk about temperature. So all normal body temperature actually repels some pathogen, and fever will actually repel the growth of it. So that's why sometimes when you're ill, the last thing you want to do is rampaging diarrhea. The last thing you want to do is lower your fever and take a anti-diarrheal, because that diarrhea effect is your body trying to eliminate out the pathogens, and that fever is trying to prevent the growth of pathogens. But again, we always obviously have to worry about the fever getting higher and so high that you can get brain damage. We don't want to do that either. So low pH, again, so your acidity of the stomach will kill a lot of our ingested microorganisms, but not all. And then we have different chemical mediators that can be involved. So, and we're going to talk about interferons and complement and things like that, that really and lysozymes that will cleave the bacterial wall, things like that. So we'll get into all of those as well. Our phagocytic barriers, so cells will actually endocytose our foreign entities. We can see monocytes, macrophages, and neutrophils that will actually phagocytize bacteria. And that's basically they create their pseudopods and form a phagolysosome, and they cause an oxidative burst to actually kill the bacteria that has been engulfed. And then inflammatory barriers. So, you know, as we get an infected cut. So if anybody's ever had an infected cut, or even, you know, ingrown hair or a pimple, you get that area of pus and fluid. Well, that's your protective. All of that in there are your white cells, your serum proteins, and things like that that help induce an immune response and help protect your body. So yeah, it might be gross looking, but it's doing its job. And then other things that can be there, your lactic acid and sweat, mucus secretions, flushing action of urine, you know, things like that. Again, normal flora, we talked about that. Your internal defense mechanism, so basically your phagocytosis again. We also have something called acute phase reactants. So these are basically your C-reactive protein, complement, things like that, that are actually enhancing phagocytosis. That's their main job. So other things we can see, haptoglobin, fibrinogen, these types of acute phase reactants, they're stimulated by cytokines, and we're going to talk about cytokines specifically. There's an entire really, really boring chapter about it, so I'll break that up into little pieces and record that one for you so you can watch them like at 5 minutes at a time. It'll be a lot easier for you. So basically these are produced by monocytes and macrophages during that. So some of these would be like your different interleukins, your tumor necrosis factors. These are cells that cause crosstalk. And you may have heard about cytokines in the news. Has anybody raise your hand and give me a reaction if you've heard of cytokines or cytokine storms lately in the news? Anyone? No? Really? Oh, Catherine has. Yeah, so the idea is that there's been some postulation about COVID actually causing like a cytokine storm in these patients, and that's what's, you know, influencing some of their really, powerful immune response to the infection itself. There's a new article on how it gains entry and the how it really messes with the bradykinin system. So it's really interesting, and it's very fascinating. If you want it, I'll send the article to you. Maybe you don't. I don't know. But we will be discussing it later when we talk about cytokines anyway. I'm not going to do any polling today because I haven't quite figured out how to do that yet. So acute phase reactants have been determined to be elevated in a patient. What does this result on its own indicate about the patient? Any ideas? You don't have to wait. You can just answer. Anybody? Well, what is an acute phase reactant?
C reactive protein compromise of A.
So acute phase reactants are they help promote phagocytosis. So typically, what are we going to see with phagocytosis?
Parasite infection. D.
Inflammation. So remember, inflammation is the. Can you hear me okay? Sorry, I just got to notice my internet connection is unstable.
Now it's okay, yeah.
Can you hear me okay?
Mm-hmm.
Okay, so acute phase reactants are part of that inflammatory response. So inflammation of an unknown cause, because we can't find the cause with just cellular defense mechanisms include our myeloid cells. So those are newts, basos, eos, monos, macros, mast cells, and dendritic cells, all of which we're going to talk about separately. One of the things that we'll talk about too, they have what's called a toll-like receptor. So this actually we discovered in fruit fly research. And basically we have a similar molecule on our white cells and some non-leukocyte cells as well. But we call them toll-like receptors. And basically the highest number of these are on our monos, our macros, and our neutrophils. So each of these will recognize a different product or different microbial product. So this is actually going to help promote phagocytosis as well. So once we have that receptor and we bind to a particular substance, we can stimulate phagocytosis. The cells will produce cytokines that are going to enhance inflammation because those cytokines are going to tell more cells that there's something wrong here. And it's basically like sounding the alarm. Just like in your house, when one fire alarm goes off, they all go off. Okay, but it talks through the electrical current. Ours talks, well, if you have it done right, ours, our cells talk through the cytokine current, if you will. So which of the following are components of the innate natural immunity?
C.
Innate natural, your T helper cells.
C.
Anybody else?
C.
Go with C, and C it is. Remember, these are going to innate, your natural immunity. You're born with that. Okay, it's always going to be the same. Your macrophages are going to do the same over and over again. Your T helper cells, your B cells, are all involved with immunoglobulin or antibody production.
Okay.
So your phagocytosis, so this is just, we'll be talking about phagocytosis, and I cannot wait to do our first immunology lab, because this is the phagocytosis lab, where we actually get to see our own white cells engulfing bacteria. It's super fun, legit, and then I let you take pictures of it on the document, on the microscope cam, so then you have pictures of your own cells doing their job for the rest of your life. It's very cool. So basically, first you're going to have that physical contact, then you're going to have the outflowing of the cytoplasm, then you're going to create a phagosome, okay? So basically, that's just, you have your engulfed bacteria or foreign entity and cytoplasm surrounding it. Then what's going to happen is that engulfed bacteria in that little phagosome, okay, it's like a little bubble, it's going to fuse with a lysosome. And basically, those are your granules within it, and it's going to create a phagolysosome. And then what happens as the bacteria is digested, we have like basically what's called an oxidative burst that will kill and destroy whatever it is that we engulfed. But you have to be aware that this does not successfully happen all the time. We cannot necessarily kill everything that we phagocytize. There are some bacteria and other things that are very resistant to that. One thing that has a hard time is our encapsulated bacteria. I mean, the bacteria was smart enough to create their own capsules, so they survived and the dinosaurs didn't. You know, sometimes we don't have a shot. Other things that we can see are chemotaxis, and basically this is where we have somatic cells and other bacteria and things will actually direct their movements according to certain chemicals in the environment. So that's where these cytokines come into play, where they can help kind of like direct them. It's almost like when someone who is visually impaired is trying to cross the streets, the signs are audible and you can hear, so they know to cross the street by following the sign of that crossing signal. So basically, when we look at phagocytosis itself, typically it's anaerobic glycolysis, and we're going to talk about all of that when we talk about cell function. But basically, when you have phagocytosis, we have this respiratory burst, so we get more energy, so the cell can do what it needs to do a little faster. If anybody's ever watched a video of phagocytosis, this is not, and actually, you know what, I'm going to try and play one for you because it's not that slow of a process. You know, chemistry nightmares here, your hexose monophosphate shunt. We also use that as well. So that's where we have the NADP to the NADPH. So then what happens is this will actually donate an electron and in this process. So if you have NADPH with that extra oxidase, with that extra electron to oxygen, you'll basically end up creating this process called a superoxide or a radical. And basically this is really toxic, but we can also make it even more lethal. So your body can actually convert it to be able to do that. So that's when you take hydrogen ions to this product, and it basically becomes a superoxide dismutase that converts that superoxide to hydrogen peroxide, and basically this will end up getting hypochlorite ions. So all of this with myeloperoxidase and everything like that, you get your oxidized killing. And again, we're going to talk more about this when we talk about cellular function and everything like that. But all of those substances that these cells produce will kill whatever is in the phagocyte at the time. That's just a reminder of your hexose monophosphate shunt. I do not ask about these in the quiz, but you do need to know that here in this process, your hexose monophosphate shunt, NADP oxidase, your superoxidase, your myeloperoxidase, and then you get your end product. So you need to know the individual chemicals within that process. Opsonization, you can also visualize this as it optimizes phagocytosis. So opsonization optimizes phagocytosis. So basically opsonins are proteins that attach to a foreign substance and kind of get it ready for phagocytosis. So it's kind of like making your food look really fancy on a plate to serve to your guest. You're prepping it for those phagocytes to eat it. So things like C-reactive protein, complement, and antibodies are opsonins, okay? So opsonins optimize. That sounded like Transformers. So what's the name of the process by which phagocytic cells are attracted toward a substance such as complement fragment C3A? Anybody know?
C.
I know it's going over, but it's done.
C.
Chemotaxis.
Mm-hmm.
So cellular defense mechanisms will include lymphocytes, macrophages, mast cells, dendritic cells. So we can see inflammation that has both cellular and humoral mechanisms involved. So our 4 cardinal signs of inflammation: redness, swelling, heat, and pain. So the major events that occur within inflammation. So number 1, you're going to get increased blood supply to the infected area. So if you have anybody ever gotten a vaccination or shot or something where it got really red and hot, anybody? Like every time I get a flu shot, I get all red and hot for just a little while. Well, guess what? That's awesome, because you want inflammation at that point, because what happens is you increase the blood supply, you have capillary permeability, your white cells will come to the area, and then you'll have macrophages to the area, okay? And in this whole process, okay, number one, we'd want to phagocytize this, but we have things called antigen presenting cells that take whatever foreign entity that they've seen, and they're going to present it to B cells, okay? And the idea is once we take whatever we've seen and we present it to those cells, they're going to start making that antibody. So that hot process is your body having an immune response that you want to see when you get a vaccine. If your body doesn't respond like that, it's either a non-virulent pathogen. So the flu shot happens to be one of those that a lot of people react very strongly to. And the reason being is we've seen so many versions of it, we recognize part of it. So that's why we get a bigger, faster response than, say, a tetanus shot, okay? Because we only get that, once every 10 years, if that. So neutrophils themselves are the primary cell that are involved in that acute inflammatory response. Typically, it takes about 24 to 48 hours for them to get there. And basically, this migration of macrophages and monocytes will come about 16 to 48 hours. And that's about the peak of it. So in this case, they're going to try and get rid of whatever is in that area as best as possible, and then trying to get the tissue to heal and not have a chronic inflammation. Because as you get chronic inflammation, the scar tissue builds and builds and builds and builds and builds. And we don't want that. The C-reactive protein is the most commonly measured acute phase reactant. So we have the high sensitivity CRP. Those of you that have been in the lab or work in the lab, you see CRPs come through all of the time, and they're really a good indicator of acute inflammation. We should be doing a lot more of these and less of the SED rates. But a lot of the times people, the old school docs, and I know they used to do this at Brockton all the time, would still use those SED rates because if that SED rate was elevated, they could admit based off of a SED rate. Things are changing though. So which of the following can be measured as an indicator of inflammation? Anyone?
E.
What?
E.
E? I'm gonna go with E. Correct. Absolutely. So I'm not going to spend too much on this. I just wanted to kind of introduce the host parasite response, because we will be talking about that in this as well. So I'm going to kind of skip over since we've gone over so much, but go through and read this. I'm not going to go through. We'll talk about parasites. So we talk about that in the chapter. I just kind of keep it in there, but we can kind of look at that after. And we already talked about that. This is always fun. Like, how can our pathogens enter? I forget. Every class comes up with a different name for this poor lady, because she gets a mosquito bite, she gets influenza, she's an IV drug abuser, she got salmonella, she also has rabies, she got tetanus, and she's got a parasite, So she's awful. But we can have entry, small breaks, lesions, ulcers, wounds, cuts, our arthropod vectors, so most commonly are your mosquitoes or your ticks, tissue damage by other organisms, and then existing pathways within the host as well. So that's how pathogens can get in. And when we start looking at pathogen response, it's really going to look at the invasiveness, the infectivity, the pathogenic potential, is it toxic, and what chemical substances are produced that can damage the host. So some of the more virulent ones would be things like Clostridium botulinum or Clostridium tetani, where they produce these toxins. So the botulinum toxin that causes respiratory paralysis and can kill you pretty quickly. That's why don't ever buy dented cans, okay? Don't ever buy dented cans, okay? I don't care that they're cheaper. Don't buy dented cans. And then Clostridium tetani actually creates a toxin called tetanospasmin, which will actually make you die like this with every muscle contracting in your body that is possible, that is there. You don't want to die that way. So that's really where we talk about things like that. And as we go through, we're going to talk about all of the things that affect humans. So we're going to talk about things like geonoses, okay? So this is where we get it from the earth. So if anybody ever watched Monsters Inside Me, a lot of these are things like fungal infections that we get out of dirt. Typically, the average Joe isn't going to be affected, but immunocompromised people are. Zoonoses is a transmission of disease from animals to man. And then vectors, we'll talk about vectors. So things like, malaria, we'll talk about Lyme disease, ehrlichiosis, things like that. And then things like fomites or inanimate objects that harbor and transmit pathogens, which I think you guys are pretty well versed because we talk about that when we talk about safety and infection control and, getting accidental needle sticks or cuts, or we touch something in the lab and then put it in our mouths, or I hope you don't, but it happens. So we're going to talk about all of these things, as we go through and look at how pathogens respond and create septicemia potentially, things like that. And we'll talk about all of that specific to our immune response and how we respond to everything else. So that's it for that one. All right, I did it. I only went over a little bit, but just a little bit. But that's, the introduction to the immune system in itself. So what I'm going to try to do when I'm recording the lectures, I'm going to try to not give you a 2-hour long lecture. I'm going to try and break them up into shorter segments. So if you're driving to work and you want to listen to one while you're driving, or if you only have 10 minutes on your lunch break and you want to listen to it at lunch, things like that. So I'm going to try, and again,
That would be good.
considering my internet is piss poor as of late, I think the recorded modules will be better, and I really want to try and leave the meet times to talking and doing cases, breaking you out into groups and coming back and reporting, our thoughts on cases. The other thing I wanted to tell you, after the first 7 weeks, we're scheduled to be in person for immunology 1 hour every Monday afternoon. What I'm thinking is we're going to take one day and we're going to do all the immunology labs in that one day so it'll be towards the end of the semester, instead of having you show up for 1 hour. Some of the labs take longer than 1 hour. And, we were so focused on, getting that 7 weeks set, we didn't really start thinking about the 8 weeks. And so I just thought of it the other day. I'm like, what am I going to do for just an hour? Let's all just come in one day and then we don't have to worry about it. Because your clinical conference on that Monday morning when you're in clinical is going to be online anyway. So there'll be a lot of Mondays that you can stay home or work instead of, coming into lab. There was one other thing I wanted to tell you, and now I don't remember. Oh, so I'm going to assume Professor George told you she retired, right?
Yeah. Send us an email.
Okay, so she is no longer the program chair. We are in the process of, I am not either, because I have all of the courses. There's no way I have enough time in the day. I mean, I'm good. I'm not that good.
You are good.
So I've got all of your classes but micro, and I have the freshmen for MED 101, plus the phlebotomy students for both those classes. So I just did my schedule. There's very little time left for me. So we're trying to hire someone new to help. So that's in process because I can't physically teach and record all day and, set up clinicals successfully. And we're doing our best to get all those, but I do want to tell you, I have a contingency plan. If the you-know-what hits the fan again, we will look at worst case scenario. We have to create some sort of virtual clinical for now, and then we do a full clinical, where you do 2 weeks hemo, 2 weeks immuno, after the spring semester. And that, again, is worst case scenario, but I wanted to tell you, like, I have a plan in my, I have like 3 plans in my head, so we can get you graduated on time, and hopefully we get a vaccine that actually works and isn't like super rushed, because that also worries me as well. So we're doing our best to kind of get these plans and have them, you know, in the back of our heads. So I wanted you guys to know that, so you can kind of breathe easy that I have plans for you. Lots of them. I probably could come up with a 4th contingency plan as well if I really needed to. But you guys were awesome last semester. I brag about you all the time. So about how well my students did. They didn't miss a live class once. So I think you guys are well prepared for it. You're enthusiastic and you really want to learn and get this done. So I'm very positive going into the semester. And I also want to lead with, if you see anything that you question, so like, I don't know who asked me, I can't see faces, so I have no idea who asked me, and you guys don't sound quite the same on Zoom as you do in person. Somebody asked me, did you move this quiz? Okay, always ask. Okay, don't be like that urinalysis class where the bookstore ordered the wrong book and they didn't actually ask me until 7 weeks into the semester. Don't do that. Yes, they had no idea, and they were like flying by the seat of their pants, but everybody was afraid to ask me, so they were using and doing the wrong assignments for 7 weeks. And they were so confused as to why the assignments didn't match up, and you know, they were putting them all in wrong, and I didn't even pick up on it because I'm like, why are they putting it in this box? Why are they working ahead? And so I was like, okay, well, this matches this, because they were matching it up with the right drop box, but they were doing things like 3 weeks early or 4 weeks early. Don't be them. Always ask, you know, I don't care. All right. Any questions before I let you go for the day?
So when we go on clinical, it will be for micro and hematology.
Yes.
Okay. And then next semester it will be
chemistry and blood bank. Adam, you too?
Yes, we're meeting for our first class on Friday. Is that correct?
Correct, 9 a.m. So it is a long day. So what I will do, like, so obviously you guys need to eat before you come. I plan on eating a big breakfast. We'll have, you know, we need to make good use of the time, because I'm worried that cases are going to spike and then we're going to have to go back virtual. So the first, like, yeah, I'm worried about it. So the first like 3 sessions, we need to take as few breaks as possible, like focus on bathroom breaks only. But I will do like a 10-minute break and we can go out to our cars and get a snack. So just be prepared to either wolf face a peanut butter and jelly sandwich, because that's pretty much my plan. You know, at least for the first, you know, 3 labs, because that way, when we get through the first 3 labs, we will have done everything but coag. So, and that's really, I really, it's going to be a lot in one day. It's going to be a very busy day, but we have to front load it because you've got to see this stuff before we get you out to clinical.
Okay.
All right. Any other questions? All right, I'll let you go. Thank you for those that stayed late that had told me they couldn't. So, and I'll be seeing you guys Friday morning.
Hey, Professor, this is Michael.
Hi, Michael.
How you doing? I'm having some trouble opening up whatever Q1H is under week 1. Every time I click on it, it closes very quickly, and it I have to try to press a button very fast. And the blood cell drawing was the same way. It opens and then closes like a terrible trick.
What are we talking about? Let's see. Sorry, I'm just going into the course space.
Yeah, so I'm under week 1. It's the it's the second tier. It's below week 1 and above blood smear prep lab. And whatever that Q1H is, I cannot open that.
You should be able to.
Okay.
All right, here, hold on. Let me try something just for giggles. See, sometimes, all right, so hit refresh on your Blackboard and try again, please.
Okay.
because it's available.
Yeah, it says diploma export test, click to launch, but I couldn't possibly click to launch that fast. It like opens it so briefly that I can't click on it.
What are you on, Michael?
I'm on a laptop.
All right, let me look. I'm going to try it myself. All right, so you can't just click begin?
It says click to launch, but I would have to get there so fast that I can't get my cursor.
So do me, all right, so one last thing. Go into the week 1 folder.
Yeah.
And click on the quiz from there.
That opens a OneDrive and doesn't actually open anything for me.
So when you're under week 1, here, let me show you. Because this, I want to make sure that anybody else might have the same problem. All right. You should see this.
I mean, mine doesn't look like that. Mine looks like what's below there. Yeah. So I see like what's in that gray box below. I don't see it the way it looks on your screen.
All right, so click to week 1 right away. And you should see this blue folder.
All right, I'm gonna try closing all of them down. It's also,
That's weird. I've never had a student say they had a problem with this. That's why I want to.
All right, so I'm clicking on week 1.
And then if you click that, you should be able to open up and see.
It just says table of contents. It's got all the things there, but I can't.
You don't have the, here, you know what? All right, let's see.
Yeah, I don't see it like your view either.
All right, let's see what you see. No one's ever had an issue before, so let's see.
I don't know if I can share my screen, but
Oh, I don't see that.
And there is a thing to launch it next, but I can't do it quick enough.
So if you go to that week 1, you can shrink your table of contents to the left. If you click on the quiz, it should pop right here, right? Because you see mine, it says click to launch.
Yeah, mine doesn't stop there though. It goes back, it just reverts right back to the week 1 folder.
That's weird.
I can open blood smear. I can open, the RBC drawing was the same way, but all
right, one last try for me. Go to the course calendar.
Yeah.
And then click on the quiz 1H in the course calendar.
Mine's working now. I just refreshed my page.
Okay, and it says go to this test.
Yep.
Okay, and then it wants me to begin the test.
Okay, so you can at least get it there. You know what I'll do is, it seems like for some reason, whatever your settings are in your computer, it's not working. I will pull the quizzes and put them in its own folder.
Okay.
And it should solve that issue. But for now, you can go to the calendar to get directly to the quizzes. I don't know why it's doing that for you. It's never, it could be there was an update and it's wonky, because this is the first time I've heard of anyone having issues with it.
So is that link just to get to the quiz?
Correct. So in the course calendar, you can link to any of the assignments by just clicking on that due date. When I took my own classes, that's how I clicked on all my assignments.
So it happens when I do the RBC drawing too. Even when I go to the course calendar, if I click on RBC drawing, right? And then I will click on go to assignment, it pops up for under a second and then goes away. And so I can't pull up the Word file.
All right, you know what? I'll move this. It seems like it's not handling the modules well.
I also have like 800 tabs open right now, so I mean, it could also
that
just
also might not help. But I'll put everything and I'll put assignments in a separate assignment folder. I'll do that.
Okay. I mean, it just seems to keep opening my OneDrive for me. But it's all stuff from
that's
like
weird.
last semester.
It's got to be a setting on your computer.
Yeah, all right. I'll try to work on like pop-ups or something, I guess.
Yeah, it could be too. If you try next time to log in out of your OneDrive, that might help.
Okay.
But I'm going to pull everything into a separate folder, and if that doesn't help, then we have problems.
All right. Well, I might bring my laptop in with me and maybe let you take a look at it if that's all right.
Yeah, that's
Okay.
fine.
I have a question. I actually do. So hold on, I wrote it down. What about our homework partner? Are we doing that Friday or?
Yeah, we'll do that in Blackboard.
Okay. And then group homework would be like the outline, case study, and then the individual is the drawing?
Yes.
And
Yeah,
how do we know what the drawings are?
Because they're assigned. They're titled and it's assigned.
I don't see that. Is it on Blackboard or in the book?
It's in the schedule. It's in the schedule and like
I see it right here. B cell and T cell.
Yes.
Okay. I think I have one more. I texted it.
Yeah, I didn't want to change all the settings because in the future we will go back to having lab groups. It takes a very long time to change all those assignments. So that's why I want to leave it the same because we copy the course space.
Okay.
What were you, Kaylee, what were you just talking about? Where did you find it?
It's in the syllabus, the assignment schedule.
All right, I'm in the syllabus now.
And the course schedule and assignments.
Oh, and then our lab, not the report, oh, you sent that. You did send that. It's not on Blackboard. You sent it separately.
So here, I'll show you.
On Remind.
Hold on.
Are you able to post that on Blackboard or?
Yeah, it's already on Blackboard. If you go under course syllabus, okay, can you see what I'm seeing? If you go under course syllabus, it's schedule and assignments.
Oh, I'm talking about the lab, like our actual lab.
Oh, they're all on, oh, the labs themselves? Yeah, no, they're all in here. If you look in each week, like they're just, sorry, it's gonna get loud because my parents are home. So if you look under each week, it has your lab. So there's your blood smear prep lab. We're just doing them all. So there's the blood smear lab. So they're all in there, but we just, we're doing them ahead of schedule on purpose.
Okay, now for the serology, there is no lab. I don't see that.
So here, I'll show you. Get back to serology. So we don't have a lab until week 3.
Okay, that's why. All right.
And then if you go to week 3, you get your phagocytosis lab, and it opens up with your phagocytosis lab.
Okay. All right. Thank you.
You're very welcome. And if you have any other questions, feel free to reach out, send me a remind, do all of that. Shh, Knox. They get excited when everybody gets home. Doesn't matter if it's 2 minutes or 5 hours. So any other questions before we finish up?
That's it.
No, I think I'll just
All right.
reach out to you on Friday. And
yeah, well, and anything else, if you have any other questions, shoot me a message, or we can kind of go over whatever we need to on Friday.
Okay.
My first outline I wrote in pencil by accident. Can I still submit it or should I rewrite it in the pen? Okay.
Please don't rewrite.
All right. Thank you.
Precious time.
Friday. Bye.
All right, bye guys. It's great to see you, some of you. Glad to have you back.
Bye.
Bye guys.